We've seen that action potentials can be actively conducted in dendrites of at least two types of neurons, pyramidal neurons in the cortex and mitral cells in the olfactory cortex. And these action potentials can either be initiated in dendrites and then propagated in a forward direction to the soma, or action potentials can first be initiated in the soma and then back propagated into the dendrite. Well, the fact that these action potentials occur in dendrites can have profound effects on the short and long-term properties of the dendrites. First, these action potentials can function to actively amplify synaptic potentials that are generated in synapses. It's been seen in other experiments that in certain neurons, in pyramidal neurons, we also see that action potentials are initiated first in the dendrite and then propagated towards the soma. Propagation doesn't have to continue all the way to the soma. It can be localized to a branch of a dendrite. But whether it is propagated just a short distance or all the way to the soma, these active responses of voltage-gated channels can function to amplify synaptic potentials. When action potentials are generated in dendrites, either through forward propagation or back propagation, these depolarizations will, of course, open up sodium channels as well as voltage-gated calcium channels, and the opening of these channels will alter the membrane resistance, the membrane conductance, and this in turn will lead to changes in the membrane time constant and length constant, and this will have important effects on the conduction of synaptic potentials to the soma. In addition, if voltage-gated calcium channels are activated by these dendritic action potentials, this will cause an influx of calcium into the dendrite, raising intracellular calcium, and this could lead to transmitter release, and indeed there are examples of synapses from dendrites onto other dendrites, and from dendrites onto axon terminals, and the activation of dendritic action potentials could trigger the release of transmitter at these synapses. And finally, increases in calcium in the dendrites and in dendritic spines as well can lead to long-term changes in synaptic strength and to synaptic plasticity. We've already discussed one example of how dendritic action potentials can be modulated. This is the example of the mitral cells, where activation of inhibitory synapses and hyperpolarization of the cell leads to a change in direction of propagation of the dendritic action potential. Dendritic action potentials can also be modulated by channels expressed directly on dendrites. And a very nice example of this is the effect of A-type potassium channels that are expressed in dendrites, and their effect on the propagation of dendritic action potentials. If one records the currents from potassium channels both in the soma and in the dendrite, as is shown in panel B, shown in the upper right, we see that if we isolate the current generated by A-type potassium channels, which is a transient current, as you'll recall, the A-type currents recorded from the soma are quite small, whereas the currents recorded from the dendrite are much larger. If we look at the amplitude of these currents as a function of distance along the dendrite, we see in fact that as we patch onto the dendrite at increasing distances from the soma, the amplitude of this A current increases. So the density of these A-type potassium channels increases the further from the soma we are. Now this differs from delayed rectifier potassium channels, which is a sustained potassium current, and that's shown in the open circles in the bottom graph, and we see that the amplitude of this current doesn't change as we move out along the dendrite, demonstrating that the density of these channels is relatively uniform. So we see that A-type channels are expressed preferentially in dendrites, but not only this, we can modulate, and there are mechanisms for modulating the strength of these A-type channels by phosphorylation. And indeed, A-type potassium channels are phosphorylated both by pKa and by pKc, and phosphorylation leads to a decrease in the open probability of these A-type potassium channels. So we have a potassium conductance that's expressed in dendrites, which can be modulated, and as we'll see, this can have an important effect on action potential propagation in dendrites. The following experiment was done to assess the effect of A-type potassium channels on action potential propagation in dendrites. Recordings were made in brain slices, and specifically the synapse between CA3 and CA1 neurons in the hippocampus were examined. So recordings were made from the CA1 pyramidal neuron, and the recordings are shown in the traces on the right. Under control conditions, the Schaefer collaterals were stimulated to activate synapses onto CA1 neurons, and a synaptic potential shown in the smallest trace was recorded. Then the A-type potassium channels were blocked by addition of the ion 4AP, which selectively blocks A-type potassium channels. So when 4AP was introduced, initially we see that the EPSP is increased in amplitude. This is 10 minutes after introduction of 4AP, and then 15 minutes after introduction of 4AP, the EPSP has gotten larger still, and we now see an action potential superimposed on the EPSP, and this indeed is a back-propagating action potential. So what we've seen is that the A-type potassium channels normally represent a large enough potassium conductance in the dendrite to block the conduction of back-propagating action potentials. But if one reduces the conductance of these channels, in this case by adding 4AP, then this reduces the potassium conductance in the dendrite and allows action potentials to be back-propagated into the dendrite. Well, one doesn't have to add a potassium channel blocker to see this effect. Normally, under physiological conditions, as the cell is depolarized by EPSPs, this will inactivate the A-type potassium channels, leading to a reduction in potassium conductance and to larger back-propagated action potentials. As I've already mentioned, if one phosphorylates A-type potassium channels, this also reduces their open probability, and indeed norepinephrine and acetylcholine inputs onto pyramidal neurons leads to phosphorylation of the A-type potassium channels and to larger back-propagated action potentials. All of the examples of dendritic action potentials that we have discussed so far were generated by voltage-gated sodium channels. These are sodium spikes that are seen either forward or back propagated in the dendrites. However, calcium channels, voltage-gated calcium channels, can also generate dendritic spikes, and the prime example of this is in the Purkinje cell of the cerebellum. Shown on the left is a drawing of two Purkinje cells by Ramon y Cajal. As seen in these drawings, these cells have a very extensive and highly ramified dendritic tree. Recordings that were made from these cells shown that the dendrites express a high density of P-type calcium channels. These are voltage-gated calcium channels that inactivate, and indeed the name of these channels, P-type calcium channels, derives from the fact that they were first described in Purkinje cells. One can monitor the activity of these channels by measuring intracellular calcium levels in the soma and dendrites of these cells. And this is done by introducing a calcium indicator dye into the cell. So one patches onto a cell, introduces the calcium indicator dye, and then looks at the fluorescence of the indicator dye, which tells one the concentration of calcium in the cell. And so this is shown in the panels on the right. The top left panel shows a picture of the dye introduced into the soma, and you can see that it also goes into the dendrites. And then the remaining panels B through I show the fluorescence given off by the indicator dye when the cell is stimulated, and one sees very nicely, starting in panel C, that calcium increases occur in the dendrites, and this is due in fact to a depolarization of the dendrite and the generation of a calcium action potential in the dendrites that propagates throughout the dendritic tree.